Hello everyone, this tutorial will go over confining cells between layers. What that means is if you have two surfaces in Blender, you can have BlenderNEURON twist your cell sections so it stays between those two surfaces. You can use this as an example if you have a 3D scan of a brain region with layers to make sure the dendrites of your cell stay within a particular layer. If you had a whole network of cells, you could confine different cell types to different layers. That's actually what this model of the olfactory bulb does. It uses scans of layers in the olfactory bulb and confines different mitral, tufted, and granule cell dendrites to different layers using BlenderNEURON. Then, like this model, you could use the forming synapses by proximity feature to create synapses based on the resulting locations. That's just one way you could use it. This demonstration is going to be a bit more basic, but let's get started. As usual, if you haven't used BlenderNEURON before, check out the installation tutorial from blenderneuron.org/tutorials. Make sure to first load your model in Neuron and start BlenderNEURON. Here's my terminal on the screen if you want to copy the steps. The link to samplecell.hawk, which I will be using, will be in the description of this video. Then navigate to the BlenderNEURON tab, which again you can get to by pressing on this arrow on the top right. And the Confine Between Layers section will appear once you import your cell by pressing Import Cell Groups to Blender. We're going to need objects to confine the cell with. If you have a file for a 3D object, you can import it by selecting File and then Import and selecting the file type. But you can also add objects in Blender by pressing Shift and A, which will bring up this object menu, and you want mesh objects, so hover over mesh, and I'll select a plane. I'll adjust the dimensions of the plane by going to the item tab. I'll increase the size to 200 meters. I'll move it down slightly, and then I will rotate the plane. Then I can press Shift and D to duplicate the plane for the other boundary, and I'll set that to z = 7. Then I'll go back to the BlenderNEURON tab and the Confine Between Layers section. I have to select objects for the start layer and the end layer. I'll select plane 1 for the start layer and the other plane for the end layer. They could also be the same object if it has multiple surfaces. Then next to Name Contains, select which sections will be confined. Asterisk means it will confine all sections with those characters in the name. In this cell model, the compartments have been grouped so that the basal dendrites are labeled dend and the apical dendrites apic, and the soma is its own group called soma. So I'm going to change this to apic to confine these longer sections, which are called apic first. And before I press confine, I'm going to hide this top plane so that I can see the cell better, and then press confine to confine the apic sections between these two planes. So now you can see that they are between the two planes. If you want different results, there's some other settings here you can mess with. You can re-randomize by changing the seed, and min height is going to be where confinement begins, and max height where confinement ends. The number here is the fraction of distance between the two layers, where 0 is at the start layer and 1 is at the end layer. So a min height of 0 means confinement begins at the start layer, and a max height of 0.5 means it ends halfway to the end layer. So with these settings, the cell will be confined closer to the start layer, which is the plane on the top here. Then max angle is the maximum angle a section can be rotated from its initial position. And max length means that any section longer than this number will be split up into sections of that length. Next, I'll confine the rest of the sections, but first I need to save these confinement results so they aren't undone. Press Update Groups with Confinement Results, which will save the results in Blender, but not Neuron yet. So now I can switch the sections to Dend, which is what those other sections are named, and press Confine again without undoing the previous results. Now the whole cell is between the planes. I'm ready to save the results to Neuron. To do that, first make sure you've saved the results in Blender with Update Groups with Confinement Results, and then press Export Groups to Neuron under Import/Export. And now the positions are updated in Neuron. That's it for this tutorial. Thank you for following along. Other tutorials can be found on blenderneuron.org/tutorials, and there will be a written version of this one on docs.blenderneuron.org under Getting Started.